So the history of the Winogradsky column is a really long and analytic project. It was invented in the late 1800s by the famous Russian microbiologist Sergei Winogradsky. This column was invented by Winogradsky as a way to enrich to grow for microbes from sediments and soils. Making a column provides an environment for the microbes to grow as a large population. This column is constructed of soil or mud from virtually any source and water from the same or different source. To these natural components are added supplemental carbon and sulfur. The key concepts are the microorganism, soil, biology, oxygen, sunlight, and ecosystem. So the methods for this column is to use a small container or a small column to put all the different ingredients such as soil, eggs, newspaper, mud, and more. Keep these four mixtures for a lot of time, about 4 to 7 weeks, and record what happened to them to these columns every week. We label our 4 bottles with carbon, sulfur, control, and carbon and sulfur. Then we added 1.5 cups of sediment to each one of them. For the carbon column, we added 1/2 cup of shredded newspaper to the sediment and we mix it very well with a mixing spoon. Then, for the sulfur column, we added the yolk of a raw egg we processed to mixing it. For the carbon and sulfur column, we added both things and then we mixed. We need 4 different control columns so we can see how different molecules can affect each bottle differently and how the process may differ. We also need a control column to see what will happen to the column if we didn't modify it from what it really is. In this case, not adding yolk or shredded newspaper. Our observations and results. Over time, nutrients and gradients grew in the columns. This is where all the microbes grow, and there are actually not a lot of them. This is because oxygen is on the top of, so all the microbes come to the top and is very visible for the human eye. There are different types of microbes, such as prokaryotic bacteria and archaea. Many microorganisms can grow in the oxygenated zone at the top of the water column. In the column that had egg yolk, there was areas of darker green, purple, and or black coloring develop over time near the bottom. These colors could make difference such as green sulfur bacteria and more. In the column that had newspaper, there were some brown and orange. Group purple non-sulfur bacteria. There was worms, snails, shrimp, and other organisms in the water. There were not a lot of them because hydrogen sulfide is toxic for many organisms. Change happened because of the oxygen of each column. For example, when time passes, less and less oxygen will organisms find at the bottom, so these organisms will come up and it will be visible. Also, change of colors. Colors will drastically change. Colors such as purple, red, orange, green, and more will appear in the columns.